Let's talk about intercellular junctions. Between cells are junctions, okay? So david7sansitheater@gmail.com, contact, consult. Alright. Don't forget to like our videos. They are always good and the best. Subscribe for more and better videos. Don't forget to share to other students and other friends to also enjoy the science, okay? So between cells are several junctions, and we are going to look at every type of junction that exists between two adjacent cells. It can be between two adjacent cells or between a cell and the basement membrane. So you're gonna see all those junctions. If we can just try to look at this, okay, you can see the normal intercellular space is around 20 nanometers, okay? So this one here, number A is zonal occludence, then B, letter B represents, we're going to talk about each and everything here, okay? So C also represents a certain type of junction. So is B, so is A. So we're going to look at everything: desmosomes, hemidesmosomes, zonal occludence, zonal adherence. So just attach to this video and enjoy. The cell membranes of the neighboring cells are connected with one another through intercellular junctions, or the junctional complexes, which are of three types. And the types of intercellular junctions, we have one-type junctions. These are also called zone occludence, or the occluding zone. In this, the outer layer of the cell membrane of the neighboring cells fuse with each other, thus obliterating the space between the cells. Okay? And such junctions form a barrier to the movement of ions and other solids from one cell to another. They prevent that movement. Right. Two, we have adherence junction. This was zonal occludence. Now this is adherence junction. In the adherence junction, the difference is that this, you can call it zonular adherence. And these cell membranes of adjacent cells are separated by about 15 to 20 nanometers wide space, which is at focal places obliterated by a dense accumulation of the proteins at the cell surface. This is a good characteristic. Obliteration by the dense accumulation of the proteins at the cell surface. So the bundles of intermediate fibers project from intercellular junctional areas and radiate into the cytoplasm. Okay, and this holds the adjacent cells at these focal places. They are of two types. One type is called desmosome and the other hemidesmosome. It is called a desmosome if the thickened focal areas are formed on both of the opposing cell membranes. It is called a hemidesmosome if the focal thickening is seen only on the membrane of one of the two adjacent cells. So this is also called a half desmosome. And adherence junctions are usually seen in the cells of the epidermis. Okay, so if we just look at this, number A is the tight junction. They become a barrier to movement of solids and ions between the two cells. You can see very tight junction. Now we have the B, which is adherence junction, which can be a hemidesmosome if it was seen on only one of the two cells, but a full desmosome or a desmosome if it is seen on both sides of the cells. So we have a focal thickening here, and another focal thickening there, and the filaments which are projecting. Then C is called a gap junction. This, we're going to look at it, okay? This is called a gap junction. As you can see, a gap junction. All right. So that is a schematic diagram for various intercellular junctions. But we'll say that this is found in the cells of the epidermis. So let's continue. Gap junction. Gap junctions or the nexus, we usually call this a nexus, are the channels on the lateral surfaces of the two adjacent cells through which the molecules are exchanged between the cells. molecules here can be exchanged in the gap junctions. Unlike the tight, each half of the channel is surrounded by 6 subunits of proteins. These are called connexins. The intercellular space between is reduced from the usual size of 15 to 20 nanometers to just 2 to 3 nanometers at such a junction. Okay, so we have connexins which form a channel. So each half of the channel is surrounded by 6 subunits of proteins, which we call the connexins. Such that the intercellular space will be reduced to around 2 to 3, that is reduced by 13 to 17 nanometers at such a junction. And the gap junctions are seen in the heart and basal part of the epithelial cells of the intestinal mucous membrane. So you can see gap junctions are usually seen in the heart, basal part of epithelial cells of intestinal mucosa. Okay, and the functions of these gap junctions are very many. Look at this. They can permit the intercellular passage of glucose, amino acids, ions, and other substances which have a molecular weight of about 1000. And they can also permit rapid propagation of electrical potential changes from one cell to another, as seen in the cardiac muscle or in other smooth muscle cells. And they also help in the exchange of chemical messengers in between cells. So these are 3 functions of gap junctions. Okay, and this is the same we saw. Now this is the gap junction. They reduced the space, which was around 15 to 20. The space is now reduced to around 2 to 3. Okay, 2 to 3 nanometers, and you can see we have the connexins. Each half consists of like 6 subunits of connexins. All right. Now the cell adhesion molecules. We can't leave this place without talking cell adhesion molecules. These are CAMs, and they are the prominent parts of the intercellular connections by which the cells are attached to the basal lamina and to each other. And the types of CAMs are here. We have integrins. Integrins, these are molecules that bind to various receptors. Those are integrins. Then adhesion molecules of IgG subfamily. Immunoglobulin G subfamily has a specific type of adhesion molecules, and through these molecules, the IgG immunoglobulins bind to various antigens. All right. 3. The CAMs, we have what we call cadherins. These cadherins are just calcium-dependent molecules that mediate cell-to-cell adhesions. Number 4, we have selectins. Selectins are lectin-like domains that bind carbohydrates. So you must know these 4 types of CAMs. We've said integrins, we have adhesion molecules of IgG subfamily, we have cadherins which are dependent on calcium, and we also have selectins which are lectin-like in nature. And the functions of CAMs are here. 1. They transmit signals into and out of the cells. Alright. 2. They play a role in embryonic development and formation of the nervous system and other tissue. Okay, so the CAMs are very important in the embryology of a person. They also hold tissue together in adults. All these functions are additions of what we came for here. Look at the functions of cell adhesion molecules. They adhere cells to each other. I mean, the functions of intercellular junctions. They are junctions between 2 cells. They hold the tissue. They enable formation of this tissue. So they hold tissue together in adults. And 4, they play an important role in inflammation and wound healing. For the wound to heal, CAMs also play a certain role there. And lastly, they also play a role in metastasis of the tumors. The tumors usually can move from one local region to another, local part of the body to another part of the body. This metastasis of the tumors, of the rapidly dividing cells, is also the comes also participate in that, okay? So that the tumor no longer remains benign but changes to go to other parts of the body. And lastly, I just urge you to like a video, subscribe for more and better videos. Click this bell always for notifications, always have a video going on, and share to other students or other science interested people to 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 also learn this science. The science is good. We're still about the human body, but we are expecting to increase to more and more and more videos. Thank you. The video 7 Science Theater.